Good afternoon and welcome to the 6th NorthShore Outcomes Research Seminar in the COVID-19 Research Series and the final seminar of 2020. Today we are honored to be joined by Dr. Richard Novak, who will be presenting his team's work and research on the COVID-19 Prevention and Clinical Trials Enterprise at the University of Illinois, Chicago. We will also be joined by an expert panel, including Dr. Jerry Krishnan from the University of Illinois at Chicago, Dr. Babafini Taiwo from Northwestern University Feinberg School of Medicine, and Dr. Kamal Singh from NorthShore University HealthSystem. Next slide. I'd like to say a thank you to our program directors and the Director of Outcomes Research and the NorthShore leadership, and also to thank our first responders and those working on the front lines during the pandemic for your service and for the opportunity to support this work. I'd also like to introduce Francesca Carlock, who is a research coordinator at NorthShore, and she will be the facilitator for this seminar, and Rachel Brewington, who's the administrator, who will also be assisting with the presentation today. So for those of you who are new to the Outcomes Research Network and seminar series, we are structured with key academic leadership. We provide support to our faculty and our researchers across the NorthShore system, and in collaboration with the Institute for Translational Medicine, our CTSA with the University of Chicago, Rush, Loyola, Illinois Tech, and Advocate Aurora. And the goals of the network are to enable uniform community access to care that is effective, efficient, and equitable, to enrich the practice of medicine, and to support key strategic clinical quality and operational decision-making. The objectives of this seminar are really to promote the flourishing of principal investigators, to disseminate our research activities and resources to investigators and collaborators across the Chicagoland, and to really promote a culture of collaboration and team science. So we would like to develop mutual awareness, to connect people through these seminars in our Incubator Hub expert panel and beyond. We'd like to bridge researchers to their peer communities across the region. And ideally, what we want to help facilitate is delivering outcomes research that demonstrates improved patient and population health outcomes and health equity for our communities. So next slide, please. Francesca?
Thank you, Dr. David, and hello, everyone. In today's webinar, all attendees will be muted with the exception of our panelists and speakers. However, there will be some opportunity for interaction. If you have any questions or comments at any point during the presentation or during the Incubator Hub discussion, please submit your communication by using the chat function as noted by the blue arrow on the screen. If you are on a computer, you can access this icon by hovering your mouse over the bottom of your Zoom screen. We ask that you give special attention to the audience of your message before you submit it. As you can see on the image to the right, you can choose to message either the panelists and speakers, which will include myself, or you can choose to message everyone on the webinar. Feel free to leave your questions and comments at any time during the presentation. And if you're experiencing any technical difficulties, please let me know by messaging all panelists and I will assist you the best that I can. I'm going to hand it back to Dr. David, who will go over the agenda for today. Next slide.
Okay. Thank you, Francesca. So the agenda for today is after our introduction, we'll have Dr. Novak's lecture, which will run until about 3:40. Or so, and following that, we'll have a question and answer session. We encourage you during the presentation to feel free to use the chat at any time to send questions or comments, and we will keep track of those and introduce those during the Q&A session. Likewise, that will be followed by the Incubator Hub expert panel with our 4 panelists, who I'll introduce after the presentation. For that, we really encourage you to look at the discussion questions and reflect on the presentation, and submit comments and questions to add to the discussion. It's really important that you do that. Feel free to put as many comments and questions as you like. We really want to hear from you. Following that, we'll do some closing remarks, and we will thank the participants and the panelists and our speaker today. So if you have any questions as well, just logistically, feel free to send those along in the chat. So it's a privilege to introduce Dr. Richard Novak, who is a professor of medicine and chief of infectious disease in the Department of Medicine at the University of Illinois at Chicago. Dr. Novak has a long and illustrious career of infectious disease research, including international work, some of the early work and development of AIDS, HIV, etiology, epidemiology, and treatment, and has been a major leader in clinical trials of vaccines and other treatments for COVID-19 research. So he's going to present on COVID-19 vaccine and treatment trials. And without any further ado, I'd like to hand this over to Dr. Novak.
Okay, I'm going to unmute. I hope everybody can hear me. I just want to mention to Sean that there's that Jerry Christian emailed me that he's having trouble getting in. His link's not working, just so you're aware. In any case, I'm very happy to be here and to talk to you guys about the prevention and treatment of SARS-CoV-2. I have, as Dr. David mentioned, I have been involved with HIV research for about 3 decades or more, actually. And as part of that, I've been doing HIV vaccine trials for the last 25 years through research funded by the NIH. And that allowed me to create the infrastructure to do vaccine trials along with other members of the networks that I belong to. And so when the coronavirus pandemic arrived, the NIH leaned on our existing sites. They looked at their existing funded infrastructure to help them move these vaccine trials forward and other coronavirus-related research. You can give me the next slide, please. So this just to introduce the idea of how of the virus and the important parts that we need to know for vaccine development is that SARS-CoV-2 is it's actually quite similar in structure to the original SARS virus, another coronavirus, and the MERS virus, both of which arrived in the last couple of decades and caused pretty severe disease in humans. They use similar means of entry into human cells, and that is that they all have a surface protein we refer to as a spike protein or S protein. It's a trimeric protein that binds to the ACE2 receptor on our cells. It has a secondary receptor, it's a protease actually, TMPRSS2, but the main one we're interested in is the ACE2 receptor for the purposes of preventing transmission. So you have this spike protein, and as I mentioned, it's analogous on all these viruses, on SARS, MERS, and SARS-CoV-2. So years of research had already gone into studying the spike protein on SARS, and so its structure was already known, and its immunogenicity was known, and there had already been attempts to develop vaccines for SARS when SARS-CoV-2 came along. And then once the Chinese had published the genetic sequence of SARS-CoV-2, it was clear that the spike protein was very analogous. So you can go on to the next slide, please. So this goes a little bit more into detail into different types of vaccines. What you can see, I hope you can see my arrow, but this is a crystal structure basically of the trimer of the spike protein, and it's what is depicted on this cartoon of the virus. And what's interesting about it is so this protein is like most viruses, particularly for the parts of the virus that attach to cells to get into your cells and cause disease. These viruses have evolved over time to protect themselves from the immune system. So there's usually a critical part of the surface proteins of all viruses that attach to the receptors to get intracellular, and the viruses need to protect that from the immune system so they can still cause infection, so they can achieve cell entry. And So the early work with the SARS spike protein had shown us that, in fact, this structure has to undergo a conformational change in order to bind to the cell receptor. And so it does that in order to keep the most important part of it, which is called the receptor binding domain, this red portion of the protein, protected so that it's not exposed to antibodies or to the immune system until the critical moment when it actually binds to the cell. So there's this research at the Vaccine Research Center at the NIH had been working with this, and they discovered that they could make some minor modifications to the amino acid sequence of the protein and get it to stabilize in what's called the prefusion structure, which exposes the RBD, or the receptor binding domain, and allows the immune system to see it. So largely, most of the vaccines that are going into phase 3 trials use a similar structure. They use either a partial component of this protein or a full-length protein or trimer with a stabilized prefusion structure so that the RBD is exposed. And for example, the Moderna and Pfizer vaccines both use that similar structure, as does the Janssen vaccine, which is approaching the end of their trials enrollment period. So what can you do? So that's the starting point of the vaccine. This is the antigen that we're really interested in. We want the body to make antibodies to this protein, particularly to the RBD. And how can we best deliver that antigen to your body to induce an immune response? There are a variety of ways that do it, and these are not novel to this particular vaccine. This is basically how vaccines are made. Some are unique, and the ones you've heard the most about are down here in the lower corner. That's the RNA vaccine. So both Moderna and Pfizer use a messenger RNA, which is a synthetic RNA sequence for the full-length spike protein with this modification that stabilizes it, and it's packaged in a lipid nanoparticle, and so that's fairly novel. I mean, this has actually been worked on for a number of years. It's not like they just came up with it. In fact, all the vaccine delivery methods that are currently being used have been in development for some time. They're also not new. So when you think that it was really an incredible feat of science that we came up with these things at such short notice, is really building on existing science and existing knowledge. So they're able to take vaccine delivery mechanisms and the previous work that was done on the spike protein to develop these vaccines fairly rapidly. So other types of vaccine strategies that are shown on this slide, the ones that are currently going into phase 3 trials, in phase 3 trials, or about to go, include actually they did an inactivated virus. That's sort of that was one of the original vaccine designs. The Salk vaccine for polio, for example, was an inactivated virus. Live attenuated viruses are being looked at, but none are going into phase 3 trials. They're still preclinical. That's like the Sabin vaccine, for example, is a live attenuated virus for polio. That would be an example. Just using a protein alone is actually being developed both by Novavax and Sanofi Pasteur, and the Novavax trial is about to begin, actually. The Sanofi vaccine, at least in early studies, doesn't look that immunogenic, and so they're going to have to make some more modifications of it before it's ready for prime time. You can use just the sequence that codes for the receptor binding domain. This is being developed. There's none that are going into phase 3 trials. This one here, H, the replication incompetent vector vaccines are being used, and examples of those are the AstraZeneca vaccine, which is a chimpanzee adenovirus, and the Janssen vaccine, which is a human adenovirus. And we'll talk a little bit more about those as we go along. And they basically contain viral DNA that's been packaged into these viral particles. They're replication incompetent, meaning they get into the cells very effectively, but they don't make viral particles. We can go on from there. Why don't you go ahead and move to the next slide. So you can see that there of these different types, the numbers here indicate the number of vaccines candidates that are in development. There are a lot of vaccines are in development by a number of different investigators and companies. The ones in peach are in preclinical development, and the ones in blue are in clinical development, but only a handful have moved on to phase 3 trials. So you can go on to the next slide. We'll show you. So I mentioned at the beginning that I was sort of asked or conscripted in a way to become part of what later became the Coronavirus Prevention Network. Again, it was a conglomeration of all the existing clinical trials networks that had been involved in prevention or vaccines. And we were asked, because we had experience and infrastructure to do clinical trials, to become part of the what became known as the COVID-PN. And that evolved over time what their mission was. When we first started discussions, it looked like there were going to be 9 vaccines in trials, and they backed off to 7. And they ultimately decided on 5 vaccines to go into phase 3 trials. And we at the UIC were asked to begin with the Moderna study of the Moderna vaccine, which was the first one launched by COVID-PN. And AstraZeneca was also launched here in Chicago at Northwestern and Rush and Cook County. And these, the AstraZeneca trial is still in recruiting. The Moderna study, as you know, as you probably heard, has been fully recruited. We already have preliminary results enough that we're expecting the emergency use authorization this week. The Janssen vaccine trial is about to finish enrollment. It will be actually, the last day of enrollment will be Thursday. It'll be fully enrolled. And Novavax is about to launch. These timetables are a little bit off, but they're close. The Novavax is about to launch, I believe, this week. And the Sanofi has actually been put on hold. We can go on to the next slide. So let's talk a little bit about what constitutes vaccine trial endpoints. So for I should mention, by the way, that the Pfizer vaccine is not part of the COVID-PN. Pfizer elected to do their vaccine trial without federal support. The COVID-PN is part of Operation Warp Speed, and it doesn't do just vaccine trials. It has other prevention strategies as well, mainly monoclonal antibodies. But anyway, so vaccine endpoint. So for both Moderna and Pfizer, the number one endpoint, actually the primary endpoint, is prevention of disease. So they want to be able to, for vaccinees to have asymptomatic infection, or hopefully not get infected at all, but actually that's not the primary endpoint. Primary endpoint is just to prevent disease. Other potential endpoints are to prevent infection entirely, and these studies do have the power to look at that. Other secondary endpoints is, if you can't prevent disease, can you make the disease less severe? Can you prevent people from being hospitalized? And failing that, if they do get hospitalized, you can prevent them from having severe disease, putting them in intensive care or resulting in death. And lastly, and very important, is, do these vaccines prevent transmission? So the vaccine trials that have been done to date cannot answer this question. Do they prevent transmission? Because to know that, we would have to be able to look at viral shedding quantitatively, and then also look at subsequent transmission cases through contact tracing to see if people who did get infected were prevented from transmitting it to somebody else. So, so far we don't have that information. We do, I mean, the studies that have been reported on so far really are looking at prevention of disease, and we'll get into in some detail how we know that that was effective. You can go on to the next slide. So let's talk first about the Moderna study because it's the one I'm most familiar with, but we'll talk about all of them since this was the one we participated in. So this study enrolled 30,000 participants. It was a one-to-one randomization between vaccine versus placebo. Vaccine is a 2-dose vaccine given 1 month apart. And they initially, the idea of enrolling 30,000 participants was because the way these studies are done is people get vaccine and then you monitor the study population to see who goes on to get coronavirus. You keep in mind, of course, we're not giving people infection. They're just going out into the world and they may encounter a coronavirus and then we monitor them. And if they have a clinical symptoms consistent with a coronavirus infection, we test them further to confirm whether or not it is, in fact, coronavirus. And then we continue to follow them to see how long they're symptomatic, how severe their illness is, et cetera. And also we look at how long they continue to shed virus after they become infected. Because we're in a surge right now, the number of cases that have been accruing in the study population has skyrocketed. And so these studies, both the Pfizer and the Moderna studies, have ended much quicker or arrived at results, I should say. They haven't ended. They're 2-year studies, but they've arrived at results much quicker than we had expected. And so to the point of submitting to the FDA, there have been 196 cases of COVID-19 in the study population. And of those, 185 of those cases were in the placebo group and only 11 in the vaccine arm, which resulted in an efficacy of 94.1%. That number may continue to fluctuate a little bit as cases continue to accrue, and they are accruing at a very rapid rate. So like I said, the study is a 2-year study, and we're continuing to monitor our participants. Part of the purpose of a 2-year study was we didn't know that that cases would accrue this rapidly. We also want to know about the durability of the vaccine. How long is the immunity going to last? One important finding of the study was that there were 30 cases of severe disease among those 185 cases, and among the 196 cases, and all 30 of those occurred in the placebo arm. So the vaccine essentially is 100% effective in preventing severe disease. They also looked across different age groups, races, and ethnicities, as well as gender, and they found that the efficacy was somewhat similar. More recent data showed actually, and as these cases accrue, is that for the older population greater than age 65, the efficacy is somewhat less. It's in about the 80s percentile range. That number again may fluctuate again. So as you look at these smaller subgroups of age, race, and ethnicity, which are not the whole population, the power to look at efficacy is less, and the confidence intervals are larger. So as more cases accrue, those numbers may continue to fluctuate. In any case, we're expecting that this Thursday, the FDA, or the VRBPAC committee, which is the committee that reports to the larger FDA, is going to meet. That's going to be a public meeting. I think it's live streamed if you want to watch it, where they'll discuss the merits of the Moderna vaccine. And it's usually a day or two later that the FDA makes a decision. Last week, when Pfizer went through this process, the FDA stepped up their timeline slightly. So by evening of the next day, they made the recommendation to recommend the emergency use authorization. You can go ahead to the next slide. So this is the Pfizer, the results from the Pfizer vaccine. So again, the Pfizer vaccine and the Moderna vaccine are nearly identical. They both target the same spike protein, as I mentioned. They're both mRNA vaccines that are packaged in lipid nanoparticles. And in their study, they enrolled over 43,000 participants worldwide, and they had the same endpoints, and they found 95% efficacy in preventing COVID-19 disease beginning 28 days after their first dose. It's a 2-dose vaccine given 3 weeks apart. I mean, as you know, people are already getting it, probably in your medical centers, they're already getting it today. So they had 170 confirmed cases, and 162 occurred in the placebo arm versus 8 in the vaccine group. They had 10 severe cases. 9 of those occurred in the placebo arm, and 1 was in the vaccine arm. Again, they also reported that efficacy was consistent across all ages, genders, race, and ethnicities, geographic areas, and they reported the efficacy in adults over the age of 65 was over 94%, which is striking. So one of the most amazing things to me about these 2 studies is these studies were looking at very similar, nearly identical vaccines, but they were done totally independently in very large populations. These are huge studies, 43,000 and 30,000 people. So nearly 75,000 people were studied, and they came up with nearly identical results. That is not a coincidence. That is just a testimony to the science, frankly, that these vaccines, if you test them in different populations and do different studies, you still get the same result. That's an incredible result, frankly, and should give a lot of people confidence that the results are real. It certainly does for me anyway. There are no serious safety concerns for either of these vaccines. And most of the safety issues were what we call grade 1 or grade 2. People who get the vaccine tend to get a low-grade fever if they get any reaction, and a fair number of people do. I'd say 10 to 15, 20% will have something of the range of low-grade fever or body aches. Headache is common. Fatigue is very common when it occurs. And they last for 24 to 48 hours max. We noted in the Moderna trial that the second dose of the vaccine was more likely to cause those adverse reactions than the first dose, not unlike the shingles vaccine, if anybody's had that, Shingrix. You can go ahead to the next slide, please. Let's talk about the AstraZeneca vaccine. Now, this is a vaccine that was developed at Oxford by scientists. It's okay. Anyway, it was developed by scientists at Oxford, and they had been working on the chimpanzee adenovirus for some time as a vaccine vector. It's really cool. So it's a vector vaccine. They use the spike protein DNA. It's a full-length spike protein. They use a TPA leader sequence, which induces greater immunogenicity. And one of the great goals of these researchers was to develop a vaccine that could be used worldwide. It was cheap, easy to reproduce, easy to transport, and could be used in developing countries. They sought out a corporate pharmaceutical for a partner, and they landed on AstraZeneca as their partner to help develop the vaccine. And they've gotten some bad press, unfortunately, because they had some bumps along the way, which you've probably read about. So what actually happened was they initially started 3 separate vaccine trials. One was in the UK, and then there was another one in Brazil and another in South Africa. And initially, they were using different manufacturers to make the vaccine product in the UK than they were in the other countries. And after they'd given the first dose of vaccine in the United Kingdom, they realized that the way they're quantitating the virus in that manufacturing site was inaccurate, and actually people were only getting a half dose rather than a full dose that they had intended. So they corrected that, and they gave the second dose as a full dose. So that was perhaps serendipitous or perhaps not, but it turned out that in that study, the efficacy was 90%. So at first they touted that that was something that was planned, but actually it wasn't. The other issue they had with their design was that they started the study in Brazil and South Africa, and initially they wanted to test it as a single dose vaccine. But after the first dose, they decided that the immunogenicity was not that great, and they decided to give a second dose. But the results they reported were that people got 2 doses at least 1 month apart. But in fact, some people got dosed as much as 3 months apart. And so it wasn't really a homogeneous population. And they ended up with only 62%. And by the way, they both got 2 full doses as opposed to a half dose. So that study showed 62% efficacy in both Brazil and South Africa. And so they reported a 70% efficacy when they averaged it together. But really, I think it's a little bit comparing apples and oranges here, and it's not really that clear if that's what we're going to see. There's still the trial underway in the United States, and maybe we can hear more from Babafemi Taiwo, who's conducting this trial, when we get to the discussion, but he can give us more information about it. But another caveat about this study was that there was a case of what looked like transverse myelitis in one of the phase 1 recipients early on when the phase 3 had started, and they put a hold on the study worldwide. They restarted it in other countries, but in the United States, the hold remained for a much longer period of time. It turns out that there have been so far 3 cases of neurologic disease. One was in a placebo, one appears to be a case of multiple sclerosis, and then the original transverse myelitis, and supposedly that person fully recovered. So there's that. I think that one of the concerns that the FDA has had was that AstraZeneca was not forthcoming about the original case of transverse myelitis when they were reporting to the FDA, and they somehow neglected to tell them about it. And the people at the FDA found out by reading the newspaper, and they were miffed, to say the least. So you really have to be very transparent when you're talking to the FDA. I think that that's the key here. They do this study does have some information on asymptomatic infection, and they were, interestingly enough, in the low dose standard dose arm, they were able to show about almost 60% efficacy against asymptomatic infection, but they saw only less than 4% efficacy in the standard dose group, and that's a concern if that's real. We don't have any of that information in the other studies because that's not been looked at. Hopefully they'll get that information forthcoming, but the concern there is that we want the vaccines to be able to prevent people from passing on the virus to others, and so if it doesn't prevent asymptomatic infection, it might not be lowering the amount of viral shedding as well, but we have no information on that. Okay, you can go to the next slide, please. So the next and the last vaccine I want to talk about is the Janssen vaccine, and then I'm going to stop. And so Janssen is a human adenovirus type 26 vector vaccine. Now this vector had been something that's been under development for some time. We actually have an HIV vaccine trial underway with the same vector. And there is a licensed Ebola vaccine using the same vector. So much work had gone into this particular vector in advance, and again, they're using the NIH's spike protein modifications in for their antigen. This vaccine has some advantages in that it's a single dose vaccine. It does have to be stored at -20, but it's much easier to manufacture and also to ship. And I mentioned the trial is going to close to enrollment on Thursday. Thursday's the last day. And because of the surge, I suspect that we'll have results of this trial probably by the beginning of January, the way things are going. And then it'll probably quickly move towards an EUA as well. I think I have another slide, but can you go to the next slide? This is another study that we're doing for the COVID-PN. We're doing a large surveillance trial where we're trying to survey the various demographic populations. Here in Chicago, we'll enroll about 4,000 people, but it's part of a larger study of almost 100,000 people in the United States, mainly looking at serology, but also testing for virus to try and get an idea what the prevalence of COVID-19 is in the U.S. Is that the last slide, or do I have one more? This is probably an important issue. I'm just looking at my watch, but so there's a lot of issues about who should be vaccinated, and you've all looked in the news, and this has been a hot topic lately as the vaccines have become available, of course, and we're already in the process of vaccinating first-line healthcare workers, and but there's a lot of discrepancies, and depending on whose guidance you use. Some people feel like seniors and nursing centers should be first line. Others feel that first responders need to be in the original group. Individuals with comorbidities, ethnic minorities are considered people who are at higher risk, who are acquiring the disease at a higher rate and having poor outcomes, mainly because of social justice issues and access issues. But other people that are being considered are people who've had SARS-CoV-2 infections but who have waning immunity. The current guidance actually is to vaccinate everybody regardless whether they've had the disease or not. And then, of course, we think mostly about the United States, but of course, this is a pandemic, and we have to think about how we're going to vaccinate the rest of the planet. Can you go on to the next slide, please? All right, I'll just finish with this then and say that we're also doing monoclonal antibody studies. You already know that these, both the Regeneron and Lilly monoclonals have been given EUA status for treatment of early disease, but we're conducting trials for prevention as well. And the Lilly product is part of COVID-PN 3501, which is studied to see if it can prevent infection in nursing home settings. That study is actually fully enrolled, and we're just in follow-up. COVID-PN 3502 is with the Regeneron monoclonal antibody cocktail, and that's to look to prevent disease in households. So we identify an index case, and then we approach other adult members of the household, see if they will be randomly assigned to antibody or placebo, and see who gets coronavirus infection. I think that really is my last slide. You can move forward.
Great, thank you, Dr. Novak. This is truly a major contribution to public health and definitely impactful for the pandemic. So at this time, we can take a few questions from our listeners. As stated earlier, please input any of your questions in the chat box and address it to the panelists and attendees so everyone can be involved. So one of our first questions looks like, Dr. Novak, were there any pregnant or breastfeeding subjects included in the Moderna trial?
Well, so actually they were excluded from the study, but it turns out that there's some people who became pregnant. I can't remember, it's a small number. Nothing adverse has been reported from them, and the guidance actually is to go ahead and vaccinate pregnant or lactating women with the vaccine, to not use that, to not withhold the vaccine. But there is a registry to follow pregnant and lactating women.
Okay. There's another question. So what was your experience with recruitment numbers for your vaccine trials? And how was this with regard to minorities and under-resourced populations?
So we got a little bit of a late start in this. We didn't begin enrolling till August, when the study had opened in July. And we enrolled, and we enlisted our colleagues at the University of Chicago to help us as enroll. So between the two of us, we enrolled, at UIC we enrolled about 300, and U of C enrolled about 125 in Moderna.
Great. Looks like.
And you mentioned minorities, and 75% of our cohort are ethnic or racial minorities, actually, at the University of Illinois.
Excellent. It looks like there's another question concerning subgroups. So for the Pfizer and the Moderna vaccines, did they have any subgroup analysis on how effective it is for immunocompromised groups, such as patients on chemo or the elderly?
People on chemo were excluded from the study, and the elderly actually were focused on, and the intent was to have about 40% of the cohort be over the age of 65. Or be under 65 with comorbidities. And as I mentioned, right now, what they're submitted to the FDA is about 80% efficacy for people over the age of 65. That's a moving target. 2 weeks ago, it was 94%. And as more cases have accrued, it's dropped a little bit. That may go up again, or may go down further as new cases are added.
Great. There is another question on targeting racial and ethnic minorities. Did you have any specific community engaged strategies to recruit racial and ethnic minority populations for the Moderna trial?
Well, we did, although I have to say that there was so much interest in the vaccine trial that by the time we began enrollment, we had 8,000 people in registries that we could invite to be in the study. And so we selectively targeted minorities from the registry. A lot of the registry, though, came from the results of being in the media. Frankly, there's so much interest in this vaccine that I was bombarded by the media to talk about it, and I used that as an opportunity to put up our phone number and our website, and people had contacted us. Our phones were ringing off the hook, frankly, and we had tons of interest. We did other approaches. I mean, I did make targeted appearances in different media. They're more, Spanish language and African American focused radio and television broadcasts. And I had made other efforts to approach the church, which was not so successful, I have to say.
Excellent. That's really interesting as far as the recruitment aspect of this. Are there any next steps that have to be taken to give this vaccine to children?
Yes. So they're in the process of designing trials for children, since children were not included in these studies. What they hope ultimately is to understand what the correlates of immunity are with these vaccines. And so normally the way you do that is you compare the people who have breakthrough cases to the ones who were protected by the vaccine, you compare their immune responses, and you can better understand which immune responses are necessary to confer protection. There are so few breakthrough cases, they might not have enough to do that analysis properly. More may occur as time goes on. But the advantage of having that, then let's say you need to have a certain titer of antibody to reach a point of being adequately protected. Then you can do what's called a bridging study with children, where you just give the children the vaccine and you measure their antibody titers and see if they achieve the titers that you need, rather than doing a clinical endpoint study where you wait for the children to get sick. So I'm not part of the design of those studies, but those are the kinds of thinking that are going forward.
All right, there are so many questions that are coming up. Are you able to comment on allergic reactions that might occur with this vaccine? There have not been any major reactions to the Moderna vaccine reported yet. That doesn't mean it won't happen. There were, I'm sure everybody reads the paper, and so they know, or watches the news and knows that there were a couple of anaphylactoid reactions to the Pfizer vaccine when it was first rolled out in England. That was unexpected because they weren't seeing that in the clinical trial either. But there's a warning about people who are severely atopic or people who have a lot of allergies and have to carry an EpiPen, that perhaps they need to be observed more closely, or perhaps not take the vaccine. But I think that those are really sporadic and pretty rare responses. Okay. Another question. Do you think that with the deployment of EAU approved vaccines that we will be able to achieve sufficient number of patients vaccinated for herd immunity?
Good question. And I would say that we've got a serious problem with vaccine shortages. I mean, there's we're not going to have enough vaccine to vaccinate the population for some time yet. We need more of these vaccines approved. I think the Janssen vaccine and possibly the AstraZeneca vaccine both will contribute a lot towards having adequate vaccine supply so we can vaccinate more people. If we're relying on just Moderna and Pfizer, it's going to be months before we can get there, and I'm not sure we can really rely on just those two. So we need more vaccine products, more companies making vaccine, and I mean, the best estimate now is that maybe by summer we will have enough vaccine that we have vaccinated enough of the population that we could potentially achieve herd immunity, but it also is very dependent on people being willing to be vaccinated. And that's a major issue right now, and we need to talk specifically about that, because there's a tremendous amount of distrust in the process, and it differs by different racial and ethnic groups. Many people feel like this vaccine has gone too fast, and then they're afraid of it, and they don't trust that it's effective. I mean, there's so much disinformation. Some people don't believe that coronavirus is real. So there are a lot of barriers to vaccinating the entire population, and what we really need to focus on now is addressing those barriers so that we can see if we can get people to vaccinate, because to get to herd immunity, we need to vaccinate probably 70 to 80% of the population. And that's a huge undertaking. If we were to have enough vaccine to do that, we still need to convince people that it's something they should do.
I think we have time for one more question, and then if everybody could reach out to Dr. Novak privately by email with their further questions. The last one we're going to take is, what do we know so far about the duration of immune response post-vaccination?
Well, we don't know much. I mean, because the vaccines are not been out long. We know that they work real well to about 3 months, and what I've heard, although I've not seen the data, is that the titers, the antibody titers don't seem to be falling off much over that 3-month period of time. So if you look at that trajectory, it looks like it's going to last for a while, but we're assuming that that is the main correlate of immunity. It probably is, but we don't know that. And so that's why these studies are designed to go for 2 years. We really need to follow people for a longer period of time and see if their immunity wanes, if we start to see more breakthrough cases as time goes on. We need to find out if maybe we need to give a booster at some point along the way. We just don't know any of that right now.
Thank you, Dr. Novak. I apologize for interjecting, but so that is all the time that we have for questions. Please feel free to insert anything additional into the chat box for our presenters to review. I will now hand it over to Dr. David, and he will introduce our expert panelists for the incubator HUD panel discussion. Next slide, please.
Thank you very much. And I would like to echo Francesca's comments. Dr. Novak, thank you for a brilliant presentation. It represents really heroic teamwork and research leadership. And we're just really delighted. We've learned a lot from this presentation today. So thank you again. For our next segment, I would like to introduce our 3 expert panelists who will provide some additional insight into the implications of this research for health equity, for policy, for implementation science, as well as future translational research implications. And I'll start off and we'll introduce Dr. Jerry Krishnan. Dr. Krishnan is Professor of Medicine and Public Health, and he's also Executive Director for the Institute for Healthcare Delivery Design, and he's an Associate Vice Chancellor for Population Health Sciences at the University of Illinois at Chicago. Dr. Krishnan is also a leader in the Vaccine Core program, and we're delighted to have him join us today, and he's very active in implementation of vaccines at his institution. We're also joined by Dr. Babafemi Taiwo, who is actually going to join late because of another meeting. He'll be joining us shortly. Dr. Taiwo is the Chief of Infectious Disease in the Department of Medicine and Gene Stollerman Professor of Medicine and Infectious Diseases at the Northwestern University Feinberg School of Medicine. And I'm also honored to introduce Dr. Kamal Singh, who is Director of Microbiology and Infectious Disease Research at NorthShore University HealthSystem in the departments of Pathology and Medicine. So thank you all panelists very much indeed for joining us today. And if we could go on to the next slide. So after listening to that presentation, and for each of our seminars, we want to make sure that we explore with our expert panel, what are the implications of this research? First, for health equity. Secondly, for healthcare dissemination and implementation. Third, for policy. And fourth, for future translational research directions. So to the panelists and also participants, feel free to send more comments about these issues. Dr. Novak provided information about a number of things concerning health equity, a really impressive recruitment record in minority populations as well. But what are some of your thoughts about the implications of this work for health equity? Feel free to go ahead and speak up at any time.
Sure. This is Jerry Krishnan. I can start, and I'm sure my colleagues can add to it. First of all, thank you for the opportunity to be with you here today, and really enjoyed listening to Dr. Novak's presentation. I think there's a lot of details, and it's coming fast and furious, and it's good to have it in an organized way, such as was presented just now. I think, if we look at the epidemiology of COVID-19, I think many of us have heard, and certainly I think a lot of us have the experience in noticing that currently COVID-19, as with many other conditions, disproportionately affects those who have the least. There's a number of, both in the lay publication, but as well as in research articles that have demonstrated that individuals or neighborhoods or zip codes that are particularly vulnerable to begin with, lower socioeconomic status, higher unemployment, violence, lower education, et cetera, are the ones that are disproportionately affected by COVID-19. And therefore, even if you were to think about what Dr. Novak just mentioned, which is we now have a vaccine that, pretty much in 95% of the cases can reduce the risk of you getting COVID-19. And if it were to be applied uniformly across all zip codes, that there would be a disproportionate benefit to those neighborhoods that are actually currently disproportionately affected by COVID-19. So I think we should look at this as a grand opportunity to address in a relatively quick way health inequities. There are very few circumstances in which you can, turn around the health inequity train so quickly as the vaccine appears to show. I think one of the interesting things about the vaccine is that, at least the Pfizer vaccine, is that there seems to be a demonstrable effect after 10 days after the first dose in terms of the risk of getting COVID-19. So I would say, number one, that if we were to think about COVID-19 and who it's currently affecting, I think vaccines have an opportunity to dramatically promote health equity. Number two, I think, which relates really to dissemination and implementation, and so I'll leave it there for the other panelists to pick it up. But really, I think what we have to think about is, Dr. Novak also mentioned that those communities that are currently, disproportionately affected by COVID-19 also have more distrust of the vaccine. And so our work, I think, is cut out for us in being able to reach individuals in neighborhoods that are disproportionately affected. And that's really, I think, a critical piece of the next steps that we should be thinking about, is in order to bring the promise of the vaccine to those neighborhoods most in need, we have to think very carefully about how do we address the concerns those neighborhoods have, who are the credible messengers, for that activity, and how do we create mechanisms so that we get to the last mile where the actual, the actual vaccination gets in somebody's arm.
Thank you. That's a wonderful answer. And it seems like there must have been a lot of good work done recruiting in terms of the community engagement and developing trust at your institution. Other thoughts, Dr. Singh, on the implications of this work for health equity?
Thank you, Sean, for inviting me. It's a real pleasure to be here, listening to Dr. Novak. That was an excellent presentation, I think, of pretty much all the vaccines that are in the pipeline, including the 2 vaccines, the messenger RNA vaccines. Looks like, the rumor mill has it that the Moderna vaccine will be joining the Pfizer vaccine fairly soon in terms of getting an EUA approval. And I think that will be huge because, just to pick up on Jerry's and some of the audience members who have asked this question about vaccine availability, because we will need a very large amount of vaccine in order to immunize the population. I mean, there's, the estimate, I think, and I'd be curious to see what Jerry and Dr. Novak have to say. The estimates are, and the big question is, what percentage of the population do we have to vaccinate before we can achieve that so-called herd immunity? And I've seen many different numbers thrown up. I'd love to hear from Dr. Novak about his opinion, but one number I've heard is 70% of the population. We have 350 million people in the US alone. So that's a huge reach. And truly, as Jerry has also mentioned, this pandemic has clearly been quite skewed in its reach. It's very interesting, but when we first saw the cases evolve in the Chicago area, you could see that the individuals that were most impacted were really middle and upper middle class white Americans, many of whom had been at fundraisers, had traveled around the country, outside the country. One of our first cases were in travelers and to individuals who had been to the famous Biogen conference. and very quickly we saw it rapidly start to change. We started to see more African Americans, more Hispanics, and then ethnic communities that we didn't anticipate, but probably was a reflection of our ability to reach these communities. So we saw it in the Syrian-speaking community, the Urdu-speaking community, the Orthodox Jewish community. So we saw it in communities that we didn't think about that we had to reach out to. And then obviously, I think we all saw this, that the biggest impact of all of this was actually the elderly living in nursing homes and congregate living settings. And so I'm really glad that one of the first groups that we are going to vaccinate are the people who are living in these nursing homes, because they really, really took the brunt of this pandemic in a huge way. I think the data from Illinois alone is that maybe upwards of 50% of our deaths were in these individuals. So in a way, I'm glad that we're targeting that population. It seems also quite natural that we target the first responders, particularly the healthcare workers, as the first group that should receive the vaccine, because clearly there's no way we can look after sick patients if we ourselves are coming down with COVID. And all of us who work in hospitals, Dr. Novak, Jerry, myself, have seen huge numbers of our own healthcare workers coming down with COVID. And we are really stretched in our resources, not because we lack the ventilators, but we lack the human resource to manage those ventilators. So I think the overall decisions that were made seem very sound in terms of deploying the vaccine to healthcare workers and to congregate living settings. And then, of course, as with the other members in this committee, I think we really have to make a very strong effort to get out to the underrepresented, the ethnic communities, the African American, the Hispanic community. I think that's also a very important group that we have to reach out to. And then obviously the vaccine deniers. I think the one thing that I'm still waiting for is I know there's supposed to be a media blitz put out to kind of encourage folks to start thinking about the vaccine. Currently, the vaccine is not ready for prime time use out in the general community. But at the rate at which all of this has moved along, we will probably reach that milestone pretty quick in the early part of 2021. So I think that media blitz cannot start soon enough. So I'll stop there, but I'll end with again asking Dr. Novak to comment, if he wouldn't mind, as to what percentage of the population we do have to vaccinate, because I'd be curious about his view as well. And also moving forward with his cohort of patients, I'm also curious to know what he thinks will happen to those who receive the placebo. Will we now have to unblind them as part of health equity? And then in terms of future research directions, what he thinks about new vaccines that will be coming up the pipeline, and will we now have to change what they have to measure themselves up against? Is it sufficient now to have a placebo group, or will we be using either the Moderna or the Pfizer vaccine in the so-called control group? And these will all be sort of non-inferiority studies moving forward. And will that impact the quality of the research and the data we can collect? Thank you.
Well, so I'll address some of your questions about. So you're right on the money with the herd immunity. In the standard number is 70%. I've heard 80% in this case because of the efficacy in the 90% range. But I think either of those numbers are going to be really hard to hit for the reasons we pointed out. Or Jerry is right on them, is correct about the distrust, and that's, I think, despite the limitations of vaccine, I think we have to overcome the distrust in the products in order to get people to be willing to take it. As far as unblinding goes, I mean, this has been a hot topic that I've been dealing with every day now as we're moving forward. So it is no longer ethical to have a blinded study. We've asked a lot of the volunteers, and they are expected to be vaccinated, and they should be. And so we can't unblind them until the EUA is out for Moderna. But the plan is to move to unblind everybody in a controlled fashion and vaccinate all the placebo recipients once the EUA is released. So everybody will be moved to the front of the line who are in the study and receive placebo. We still, ideally, if this we're just interested in the science, we wouldn't unblind because we still need more information, but we can unblind people, and then essentially what will happen is we'll create two study groups. We'll have people who got vaccine early, and we'll have those who've gotten it late. We're going to get it as part of the placebo group. And then if they remain in the study, which we hope they will, then we can continue to follow them longitudinally to see if there's waning immunity over time and more breakthrough cases. And we can use that information to try and understand what the durability of the vaccine response is. At a later date, we may ask our participants to participate in a booster study if we need to give them a booster dose. In terms of placebo-controlled trials going forward, we're doing the Janssen study, and that is a placebo-controlled trial. And it's still ethical to continue that study as a placebo-controlled trial because for the vast majority of the population, they will not have access to a vaccine for several months. The vaccine's just not available. And so, in fact, the people who are in the Janssen study will probably get vaccine quicker than if they waited, because they'll either get it immediately when they're in the study, or if they end up being randomized to placebo. As I mentioned, the results of that study will probably be out in January, and they're speculating that if it's shown to be efficacious, that there will be an EUA approved by beginning of February, and at that point, that study will be unblinded, and those placebo recipients will get vaccine, and it'll still probably beat them getting either the Pfizer or Moderna vaccines by a month or two. So, but beyond that, it might be more challenging. My understanding is the Novavax vaccine is going forward as a placebo-controlled trial because it's assumed the same assumptions are being made about that study. But ultimately, what will happen is that we will have to go to, as vaccine does become more widely available, we'll have to go to comparative trials and look for non-inferiority type of questions.
Excellent. Thank you so much. Dr. Tylo has joined our panel. My apologies, Dr. Tylo, for the issues with the web link. We're delighted you can join us. And we just heard a really brilliant presentation by Dr. Novak about his vaccine and other treatment trials. And we're at the expert panel where the first question is, what are the implications of this vaccine research, including your own, for health equity? And so, sorry to put you on the spot, but if you have thoughts to share.
No, I think it really has been an opportunity to renew the commitment that the research community has to the entire Chicago population, and by extension, the entire citizenry of the United States. Clearly, like the epidemic itself showed, there are significant discrepancies in terms of risk and outcomes that disfavor persons of color. And I think that same trend was initially seen in the recruitment into the trials, which was a legacy of the distrust that certain communities, particularly Black community, has held for the research community. And I don't know if Dr. Novak mentioned it, but in fact, the accrual target of the Janssen trial of 40,000 was reached over a week ago. But there was a deliberate hold-up because the numbers of people of color, Black and Hispanic, in the trial, they were grossly underrepresented. And so a push had to be made to really make sure that the representation is adequate. And that's the only reason the study is still open to accrual today and is expected to close to accrual on Thursday. But I think we should not only acknowledge the problem, but also celebrate the response to it, because the study was held back so that equity could be maintained. So I think that testifies to the fact that there are certainly efforts being made to equalize whatever inequity exists, but for sure it will take time. And even myself, I continue to learn new approaches and new ways, and gain even deeper respect for our obligation to make sure that our health equity is at the forefront of research, and hopefully not lose the gains that we've made from this epidemic and translate it hopefully into other disease conditions such as HIV, which is my primary area of work, and also related infectious diseases.
Well, thank you very much. And Dr. Krishnan also made a comment about this being a real opportunity to reach out to under-resourced communities across Chicago, which would have, if it's effective, the biggest effect, because the inequities are so great. And Dr. Novak commented on 75%, I think, minority recruitment to Moderna trial. Is that right?
That's what we had in our site. No, overall, in the total study, it was much less than that. It was, I think there were 10% African American and about 23%, or that range, Latino or Latinx.
Okay. Well, thank you for your responses on the health equity question. And the next question is, what are the implications of this research for health care dissemination and implementation? And also, we've had some additional questions come in I would like to address. The particular one has to do with the issue of cold storage, temperature control, the vaccines, and what are the implications there for distribution. And another has to do with just the incidence of serious adverse events in the trials, whether you could comment on how likelihood that is to happen from population level. So yeah, if we could go on, Dr. Krishnan, this is really your area. Any thoughts on, I'm sure you have a lot of thoughts as to what you do, but all the panelists and at the institutions, what are kind of big picture, high level, what are you doing for dissemination implementation?
Yeah, thank you again. And I think that, so I think if we think about this, the research has, perhaps two broad phases. One is the discovery phase, where we're trying to figure out what to do. And then you have the delivery phase, which is now, how do you get it? How do you get reach? And how do you make sure that it's consistently applied, in the intended audience, so that we have maximum public benefit of the massive public investment that went into these clinical trials. So you are absolutely right. I think all of us right now, even within our institutions, as we start to receive the Pfizer vaccine and then hopefully soon the Moderna vaccines, we're now doing dissemination implementation planning for healthcare workers. So even within our institutions, we're thinking about what is the messaging that needs to come out that addresses what people want to know. And some of that might be just logistical information. Where do I go and how do I get vaccinated? But in other cases, just maybe particular concerns people might have about side effects, perhaps about whether the process was legitimate or was it somehow cut short. How do I trust the vaccine? So we have to think about the messaging, now, and we're already doing it, as I said, for healthcare workers right now, but we should be thinking about it for the community. And then beyond the messaging is, how do you get in reach into the very populations that are disproportionately affected? And for there, I think we have to think very carefully about who is most susceptible, who, if they do get infected, who are more likely to have severe disease. And so to that effect, I think the National Academy of Medicine came out with a report a few months ago. And then subsequent to that, the Centers for Disease Control and the ACIP committees have provided us some additional guidance about which populations should we be targeting or implementing initially, recognizing we don't have all the vaccines for everybody. So we're going to need to do some triaging or some phased implementation. Given this issue, I think all of us recognize that in Chicago or in Cook County, many times we're sharing similar populations across our healthcare systems. There are patients who are coming from South or the West Side, I'm sure, coming to North Shore. We have some of those patients at UIC, and I'm sure that Chicago sees patients from the North and the West Side. So I think part of this is recognizing that the dissemination and implementation process is going to need to be carefully thought out and coordinated in such a way that the messaging is consistent no matter where it's coming from. Because if we start to say slightly different things, people will focus on differences of how we're framing it, and perhaps less so on the fact that we're all making the same statements. About 3 months ago, we began a group called the Vaccine Corps Partnership, which is multi-sector collaboration of health systems, community-based organizations. CDPH, Cook County, and IDPH are now participating and regularly presenting, along with the philanthropy and a number of other organizations. So the Vaccine Corps Partnership is a forum in which we're trying to create opportunities for collaboration and cooperation regarding dissemination and implementation. And the goal is to train and provide appropriate training and support for credible messengers to different communities. And I think there are representations from all parts of Chicago in it. And currently what we're doing, and then I'll pause and have our colleagues comment, is we have actually started a series of projects in which we are deliberately going into communities and asking individuals what are their concerns about the vaccine, what information would they want to know in order to make a decision, and who would they want to hear it from? Who are the credible messengers in their communities? And we are not directly at the university doing that. We're actually working through community-based organizations to do in-reach into their own communities. I think sometimes if we come out as academics going into neighborhoods, we're going to have less of a reach than if we work with our partner community-based organizations to do that work. So I'll stop there. I know, Rick, you're aware of the Vaccine Corps Partnership. Perhaps you can mention a few words about that, and then we can open up for others.
Sure. I think it's a fantastic approach, and it's a great collaboration citywide. And I commend you, Jerry, for taking the lead on this. Recently, the CTSAs, the Clinical Translational Science programs from Northwestern, University of Illinois, and University of Chicago have all banded together to help offer their support to collaborate with the Vaccine Corps and offer our resources. And so it's an opportunity for all the healthcare institutions to be involved. They're currently engaged in the CTSAs. And it's a citywide effort and a statewide effort. And it's essential, actually. As I pointed out earlier, it's one thing to have a vaccine. We have to ramp up production, but then there's the issue of who's going to actually take it. And we need a lot of work. We have a lot of work to do to create the message, to be able to convince people that it's in their own best interest to do so.
That's really inspiring and amazing leadership coordination. Thank you very much. And we're all very proud of the Vaccine Corps across the CTSA. So I know that Dr. Singh and Dr. Taiwa are also deeply involved with implementation at your institutions. Any reflections on the other comments and about some of the challenges you're dealing with? We're in the middle of one here. Mm-hmm.
Yeah, thanks, Sean. It's Dr. Singh again. So this dissemination and implementation of the vaccine, where myself and so I'm involved in a more limited fashion, essentially dealing with our healthcare workers. And part of the paradox in all of this is, yes, we have some guidance, and the guidance is that the priority is for healthcare workers and for residents of long-term care facilities to get the vaccine first. But throughout this whole pandemic, a lot has been sort of delegated to individual health departments, individual medical centers, clinics to kind of craft out their own policies and procedures, which means that we're all scrambling to reinvent the wheel, each and every one of us. And so, some of us are a bit more consistent than others. And it's a shame that we have to continue to struggle and deal with that. It would have been nicer to have clearer guidance, because the paradox of this is that, while we were expecting a reasonable number of individuals to be hesitant about the vaccine, it's almost, in actual fact, turned out to be a Black Friday rush for the vaccine. So I get emails and text messages and calls from people who have very little direct contact with patient care, demanding that they should be on the very top of the list of individuals who should get the vaccine first. And so we have to create these prioritizations of groups of individuals who we think are at the highest risk. Maybe the folks in the emergency departments, in the ICUs, where they do so-called aerosol generating procedures, and then sort of, try and filter it down. But one of the things we were very, very cognizant of is there are individuals that do the work in our hospital that are definitely from underserved populations. So if you look, for example, at the folks that come in and clean your rooms, that bring in your food tray, they are more likely than not to be from underserved communities, the African American community, the Hispanic community, etc. So we were very cognizant of the fact that we have to make sure that they are also pushed up a little bit in the ladder in terms of access to the vaccine. And then finally, in terms of what are people most concerned about? And really, it's two things. They want to know, is this an efficacious vaccine? Does it work? And two, is it safe? That's pretty much most of the questions that we've been getting. Tell me that it works and tell me that it's safe. And on the balance, as Dr. Novak has shown the data to everyone this afternoon, it meets both the criteria. And then, in my opinion, again, and maybe this is biased because I'm a healthcare worker, but again, I think that is a group that should have been the first to receive the vaccine and is the first to receive the vaccine. Because when you look at policy decisions and what people listen to out in the community, they listen to the nurses, they listen to the doctors, they listen to the pharmacists when these groups of individuals come out and say, take a vaccine. They, I think, are the biggest persuaders of the general community in terms of acceptance rates. If the doctors, if the nurses and the pharmacists start saying things like, I don't think you should take that vaccine, I'm not sure the efficacy and the safety data is there, that's when you will meet the real hurdle to getting widespread acceptance of this vaccine.
Yeah, I think all those are very well taken. I would add that when you're thinking about the appropriate vehicle in terms of the voice that people will listen to, in addition to healthcare workers, in the Black community, for example, it turns out, I believe, that the faith community is very important. What we found was that we got our greatest enrollment of Black Chicagoans when we reached out and partnered with some of the African American churches. And it really is about, who do people believe what they say? If your pastor is telling you do this, and the pastor has helped you through certain things, you're more likely to take that up than a researcher just saying do this, right, for a variety of reasons. And so one, identifying the appropriate vehicle and also empowering that vehicle with the right information. So you can imagine that if you've decided that X is the appropriate vehicle for this particular community, they could be in that role and they could be spreading falsehood. So are we making sure that the people who we've identified as the proper spokespersons are empowered, they have the right information to disseminate? I think that's very important. And also in terms of implementation, we have adopted a similar approach to what's been described, where we're going to follow the tier system based on the likelihood of a healthcare worker being exposed to COVID. But if there are more than more people in a particular tier than available vaccines. How do you deal with that? There has to be some additional criteria like individual risk or even a ballot has come up as a possible way of ensuring equity. So all of these are important, but again, we can't lose sight of the tremendous opportunity that this moment has brought.
Thank you. This has been a very rich discussion. I think everybody should watch the recording of this if they've missed it today. We can at least address the final questions kind of in summary, quick thoughts about implications for policy beyond Operation Warp Speed, what's really should be next here. And also, if you have thoughts about future translational implications, we can go for a couple minutes. So I think people have talked about some policy implications Chicago-wide. Any other thoughts about implications for policy?
I mean, I would just add that this is a grand opportunity for some coordination and cooperation across our institutions. The COVID doesn't discriminate by border or zip code or race or ethnicity. I think that there are certain groups that are disproportionately affected, and we all serve, I think, those individuals. So I think we need to band together and make a statement that the healthcare community and the research community is going to aggressively work together in a cooperative fashion to erase health inequities due to COVID by systematically sharing best practices in order to have reach and implementation. So I would invite people to join the Vaccine Corps partnership, and if they're interested, they can look in the chat. I put the email address on there. But this is a way to start to compare notes. I think, as Dr. Singh mentioned, why reinvent a few wheels? If certain things are working, let us know. If things aren't working, let us know, so we're not repeating it and learning it as well.
Great.
I'm going to piggyback a little bit on what Jerry said, Sean. Actually, maybe if I can jump to just the future translational research directors.
Please.
You can still hear me there, Sean?
Yes.
In my short lifespan, and I consider myself fairly young, I was involved in 2003 during the original SARS outbreak in Singapore. And then I was assisting the CDC with infection control issues during the MERS outbreak. And now I'm involved in the second SARS coronavirus outbreak. And this is all in the measure of 17 years when we've had 3 coronavirus outbreaks. And probably the common thing here is these probably, again, I think it's true, zoonotic infections that, is a collision of human and animals, and there are some geographies and some politics here. But I think what it tells us that as a world, we have to start cooperating with each other. One infection that starts out in China can actually spread around the world, and vice versa. What starts here in the US can spread around the world, as we saw with the misnamed Spanish flu. So I think we need better cooperation. We clearly are starting to engage more with our veterinarian colleagues, but we also need to be prepared for the next and the next. And whether the next outbreak will be another coronavirus, who knows? But I think some of the work that's done in vaccines is going to be huge, because maybe the next thing we look at is a pan-coronavirus vaccine, so that if there's another novel coronavirus that pops up somewhere, we'll be better prepared to administer the treatment and the prevention and the vaccines. So my goal for future translational research is to see more cooperation, particularly in the area of emerging infections.
Thank you, Dr. Singh. And on that note, if we can advance to the next slide. I would like to give my heartfelt thanks to each of you, Dr. Novak, for that amazing presentation, to Dr. Singh, Dr. Tylo, and Dr. Krishnan for your comments. Very rich discussion. We're at the end of our time almost, but I wanted to make sure that people know if you have questions, concerns, or requests in a future conference or anything about this particular seminar, feel free to email me. My email is there. Also, if you could please take a brief survey regarding today's presentation to all the participants.